Welcome to the deep dive. We're here to really unpack some dense microbiological source material and make sense of it together.
Yeah, and today we're tackling a pretty notorious group, the gram-positive bacilli. These are rod-shaped bacteria, and, well, they include some really significant pathogens.
Some infamous ones, definitely. So our sources are, as usual, quite technical, medical micro stuff.
Right, and our mission really is to give you a clear framework. How do we classify these things? And, maybe more importantly, what makes them tick? Why are they so dangerous? We're talking survival skills and some seriously potent toxins.
Okay, so where do we start? What's the first big dividing line when looking at all these gram-positive rods?
It's actually pretty fundamental, comes down to survival. Do they form spores or not?
Spores, like little survival pods.
Exactly. That's the first major split. You've got the spore formers, Bacillus and Clostridium. These guys are built tough, super resistant to heat, drying out, disinfectants. They can hang around for ages.
Microbial bunkers, you called them. I like that.
Yeah. And then you have the non-spore formers, like Corynebacterium and Listeria. Still pathogens, still important, but they don't have that same level of near indestructibility the spores provide.
So the spore itself, it's not just a simple shell, is it? It sounds like complex engineering.
Oh, absolutely. It's a dormant structure, multiple layers. Inside, the core holds the DNA, but it's dehydrated. And the real key to its famous toughness, especially against heat, is this chemical called dipicolinic acid, or DPA.
TPA, I've heard that mentioned. How does that work? How does it stop something from, boiling?
Well, the DPA complex is with calcium ions inside the core. Think of it like internal scaffolding, maybe. It locks everything down the DNA. The enzymes makes the whole core incredibly stable and rigid.
So it sort of chemically reinforces it against heat.
Precisely. Boiling water might kill the active growing cell, but the spore just sits there. You need serious stuff, high pressure, high temperature, like in an autoclave, to actually break down that DPA stabilization and sterilize spores.
Okay, chemically fireproofed almost, and dried out, which is why you need special stains, right? Like the Schaefer-Fulton you mentioned, to force the dye in.
Exactly. Got to push that dye past the resistant outer layers. So that's our first split. Spores or no spores, what's the next big differentiator?
Oxygen, their relationship with oxygen.
Yep, our metabolic roadmap. And here, Clostridium really stands out. It's an obligate anaerobe.
Meaning oxygen is basically poison to it.
Right. It needs environments without oxygen, think deep puncture wounds, canned food, places like that.
Okay. And the others, Bacillus, Corynebacterium, Listeria.
They're generally more flexible. Aerobic, meaning they use oxygen, or facultative anaerobes, meaning they can kind of switch hit, grow with or without it.
But here's the kicker, you said. Regardless of spores or oxygen needs, they all seem to make powerful toxins.
That's the unifying theme, and honestly, it's what makes them such major medical concerns. These exotoxins are their weapons.
All right, let's dig into the first group then. The aeroic spore formers, genus Bacillus. You find these everywhere, right? Soil bacteria.
Ubiquitous, yeah. Usually large rods, sometimes in chains. But one species really dominates the conversation due to its, well, lethality. Bacillus anthracis. Anthrax.
The classic bioterror agent, unfortunately.
Indeed. And what's interesting is how it differs from its relatives. Most Bacillus species can move around. They're motile.
But not anthracis.
The anthracis is the big exception, non-motile. That's a key diagnostic clue. And its main weapon inside the host isn't a toxin initially, it's its capsule.
Ah, the capsule. And this one's unusual, isn't it? Not made of sugars.
Exactly. Most bacteria use polysaccharides, complex sugars, for their capsules. B. anthracis uses poly-D-glutamic acid. It's a protein, basically, an amino acid chain.
And that different chemistry makes it harder for our immune system to deal with.
Much harder. It's poorly immunogenic and resists phagocytosis, getting eaten by immune cells. It's like a stealth cloak made of unusual material.
Okay, so it's got the cloak, but then it deploys the dagger, the anthrax toxin, which is actually 3 daggers working together.
That's a great analogy. It's a tripartite toxin. 3 separate proteins that have to combine to do the damage. You need all 3.
Okay, what are they?
First, there's protective antigen, or PA. Think of PA as the delivery guy, or maybe the key that unlocks the cell door. It binds to receptors on the host cell surface.
So PA gets it to the target, then what does it deliver?
It delivers the other 2 components. First is edema factor, or EF. Once inside the cell, EF messes with cell signaling. It ramps up levels of cyclic AMP, CKMP.
CMP, that affects water balance, right?
Massively. Sky-high KMP disrupts water balance, causing fluid to pour out into the tissues. That's the edema, the intense swelling you see in anthrax infections.
Okay, so that's the scoring factor. What's the third part? The lethal part.
That's lethal factor, LS. and it lives up to its name. LF is a type of molecular scissor, a protease. Its specific target is a pathway inside the cell called the MAP kinase pathway, or MAPKK.
MAPKK sounds complicated. What does cutting that do?
You don't need the full name. Just know that MAPKKs are like crucial wires in the cell's communication network. They control things like cell growth, division, survival signals. LF snips those wires.
So it just shuts the cell down, causes it to die?
Pretty much. Leads to widespread cell death, tissue damage, and ultimately systemic shock. That's why it's so dangerous.
Which explains why the inhalational form, Woolsorter's disease, is so incredibly severe. Spores get deep into the lungs, into the lymph nodes.
Exactly. Leads to hemorrhagic mediastinitis, bleeding in the chest cavity, and often meningitis. It's often fatal.
Okay, that's the heavy hitter. What about the other bacillus we hear about, B. cereus? That's more food poisoning, right?
Much more common, much less severe, thankfully. B. cereus causes two main types of food poisoning, depending on the toxin involved.
Two types, okay.
Yeah, there's the emetic form, the vomiting form. This one comes on quick, like 1 to 6 hours after eating contaminated food, usually starchy stuff like fried rice that's been sitting out.
The leftover rice syndrome.
That's the one. It's caused by a heat-stable toxin that's already formed in the food before you even eat it. Causes mostly nausea and vomiting. Pretty similar to Staph aureus food poisoning, actually.
Okay, quick onset. Vomiting. What's the other form?
The diarrheal form. This takes a bit longer, maybe 6 to 15 hours incubation. It's caused by a different toxin, one that's heat labile, meaning cooking would destroy it, and it's produced by the bacteria after you ingest them down in your small intestine.
And this one causes diarrhea.
Yeah, cramps and watery diarrhea, more like Clostridium perfringens food poisoning, which we'll get to.
So two different toxins, two different symptom sets from the same bug. Okay, crucial question then. In the lab, how do you tell B. anthracis, the really dangerous one, apart from B. cereus, the food poisoning one?
There are key visual tests. Remember, B. anthracis is non-modal. You check that under the microscope. B. cereus is modal.
Okay, movement is one.
And the other big one is hemolysis on blood agar. That's a plate with sheep's blood in the agar medium. B. anthracis is non-hemolytic. It doesn't break down the red blood cells, so the agar around the colony stays red.
Right.
B. cereus, however, usually shows beta hemolysis. That's a clear zone around the colony where it's completely lyzed, totally destroyed, all the red blood cells. Big difference visually.
Non-motile, non-hemolytic for anthracis. Motile beta hemolytic for cereus. Got it. That's a critical distinction.
Absolutely vital. Okay, shall we pivot to the other spore formers? The anaerobes.
Let's do it. Genus Clostridium. Defined by being obligate anaerobes, no oxygen allowed, and also prolific spore formers living in soil, surviving harsh conditions.
And like Bacillus, they're known for their toxins. Real specialists. What's fascinating here is you have two species, C. tetani and C. botulinum, whose toxins cause opposite neurological effects, like perfect mirror images.
Okay, let's start with C. tetani, tetanus, lockjaw.
Right. Microscopically, it often has this characteristic drumstick look because the spore forms right at the very end of the rod. And unlike B. anthracis, C. tetani is motile.
Okay. And its toxin, tetanospasmin, causes spastic paralysis. Everything seizing up. The lockjaw, that creepy grin, the rhizus sardonicus, the back arching, opisthotonos. How does the toxin do that? Constant muscle contraction.
The tetanospasmin travels up the nerves to the spinal cord. And there, it specifically blocks the release of inhibitory neurotransmitters, chemicals like glycine and GABA.
Inhibitory. So these are the signals that tell muscles to relax.
Exactly. If you block the relax signal, the motor neurons just keep firing uncontrollably. There's no off switch. The result is sustained, rigid muscle contraction, like the accelerator pedal is jammed down and the brakes are cut.
Wow. Okay, so if tetanus toxin jams the accelerator by blocking the relax signal,
Right.
then C, botulism must be the opposite.
Precisely. Botulinum toxin causes flaccid paralysis,
Limpness.
weakness. It works at the neuromuscular junction where the nerve tells the muscle to contract.
And what does it block there?
It blocks the release of the excitatory neurotransmitter, acetylcholine. That's the main go-see signal for muscle contraction. No acetylcholine release, no signal, no contraction. The muscle stays completely limp.
Okay, hold on. That's remarkable. Same genus, Clostridium. One blocks inhibition, causing constant contraction. The other blocks excitation, causing constant relaxation. Spastic paralysis versus flaccid paralysis.
It's an absolutely stunning example of molecular precision and how finely tuned our own neuromuscular system has to be. It just shows how vulnerable it is to these targeted toxins.
Absolutely. Okay, third major Clostridium, C. perfringens. This is the gas gangrene bug, right?
That's the one. Also an anaerobe, needs deep tissue damage. poor blood supply.
Yeah.
Unlike tetani, C. perfringens is non-modal, often described as having stubby ends morphologically.
And its main weapon is the alpha toxin. Sounds ominous.
It is. Alpha toxin is an enzyme, specifically a phospholipase C, also called lecithinase. Fancy terms. But what it does is attack the fatty membranes of cells, especially red blood cells and muscle cells.
So it just dissolves tissues.
Essentially, yeah. It degrades the phospholipids in cell membranes, causing cells to rupture, leading to massive tissue death, necrosis, and hemolysis.
And the gas part of gas gangrene?
That comes from the bacteria's metabolism. As they ferment the dead and dying tissue in that anaerobic environment, they produce large amounts of gas, mostly hydrogen and carbon dioxide. You can actually feel it under the skin sometimes, a crackling sensation called crepitus.
Grim. You mentioned this one has some dramatic lab tests, too.
Oh yeah, some classics. First is the stormy clot formation test.
Okay, that name definitely paints a picture.
Doesn't it? You put that bacteria in litmus milk. C. perfringens ferments the lactose in the milk really rapidly, producing a lot of acid. The acid causes the milk protein to solidify, forming a clot.
Right.
But then the huge amount of gas it produces literally rips that clot apart. So you get the solid clot that's been violently disrupted by gas bubbles. Stormy.
Wow. Okay, and the second one, Naegler's reaction.
Naegler's reaction specifically detects that destructive alpha toxin, the lecithinase activity.
Yeah.
You grow the bacteria on an agar plate containing egg yolk, which is rich in lecithin.
Okay.
If the bacteria produce alpha toxin, they break down the lecithin, and it causes a milky white precipitate, an opaque zone, to form in the agar around the colony.
How do you confirm it's definitely the alpha toxin causing that?
You do a control. On half of the plate, you spread antitoxin antibodies against the alpha toxin before you treat the bacteria. On that half, the antitoxin neutralizes the enzyme, so you don't see the opaque zone form. If you see opacity on the side without antitoxin and none on the side with it, you've confirmed alpha toxin production.
Very clever. Visual confirmation. Okay, we've covered the spore formers, Bacillus, and Clostridium. Let's shift to the non-spore formers now, starting with Corynebacterium diphtheria.
Diphtheria.
Right? Non-spore former, non-modal. One characteristic feature under the microscope is the presence of intracellular granules that stain irregularly. Metachromatic granules or Babes Ernst bodies. But the really defining thing is the disease itself, the pseudomembrane.
Absolutely. That's the clinical hallmark.
Right.
C. diphtheria, if it's a toxigenic strain, causes this thick, gray, tough membrane to form, usually on the tonsils, pharynx, sometimes larynx. It sticks really tightly.
And it's dangerous because it can block the airway, right? Suffocation.
Exactly. It's not just bacteria. It's a whole mat of fibrin, dead host cells, inflammatory cells, and bacteria all fused together. Airway obstruction is a major risk.
So knowing if the particular strain someone has is actually producing the toxin is critical. How do you test that?
There's a specific test called the ELEC test. It's an in vitro test done on an agar plate. You place a filter paper strip soaked in diphtheria antitoxin down the middle of the plate. Then you streak the unknown Corynebacterium strain across the strip perpendicularly. You also streak known positive and negative control strains. If the unknown strain is producing diphtheria toxin, the toxin will diffuse outwards from the streak, and the antitoxin will diffuse outwards from the strip. Where they meet in optimal concentrations, they form a visible line of precipitate, an arc.
Like the Naegler test, it's antibody meets toxin.
Exactly. Seeing that precipitate line confirms the strain is toxigenic.
Okay. Our last major player today, Listeria monocytogenes, also a non-spore former. This one seems like more of a stealth operator, causes meningitis, sepsis, especially in vulnerable groups.
Yeah, Listeria is a really interesting one. Causes serious invasive infections, particularly dangerous for pregnant women, newborns, and immunocompromised people. And it has a couple of key survival tricks.
What makes it so tricky?
Well, first, it's what we call psychotrophic.
Psychotrophic.
Meaning cold-loving, essentially. It can actively grow at refrigerator temperatures, down to about 4 degrees Celsius or 40 Fahrenheit.
Wow, okay. So putting contaminated food in the fridge doesn't stop it.
Nope. It actually allows it to potentially multiply while other competing bacteria are inhibited. That's why it's such a headache for food safety, associated with things like deli meats, soft cheeses, unpasteurized milk, foods often stored cold.
Right. That's a huge issue. What else? You mentioned invasive. Does it get inside our cells?
Yes, that's its other main trick. It's an intracellular pathogen. It gets eaten by immune cells, phagocytes, but it doesn't stay trapped. It produces a toxin called Listeriolicin O, or LLO.
Listeriolicin O, what does that do?
LLO basically punches holes in the membrane of the phagosome, that little vesicle the immune cell uses to contain the bacteria. This allows the listeria to escape from the vesicle out into the cytoplasm of the host cell.
So it breaks out of jail, essentially.
Yeah, exactly. And once it's free in the cytoplasm, it can hijack the host cell's actin machinery to propel itself around and even push directly into neighboring cells, all while staying hidden from antibodies and other immune defenses outside the cell.
That's clever. Sneaky. Doesn't it also have a specific kind of motility we look for in the lab?
It does. At room temperature, around 20-25 degrees Celsius, Listeria exhibits this characteristic tumbling motility. When you look at it in a liquid culture under the microscope, it's quite distinct. But interestingly, it doesn't really use flagella for movement inside host cells. That's the actin-based motility.
So it's adapted to survive both in the cold fridge and inside our own cells. Quite the survivor.
Definitely. A very versatile and dangerous pathogen.
Okay, what a journey through these gram-positive rods. We started with that basic split spores versus no spores, then oxygen needs, and saw how those basic features connect to survival strategies and, crucially, to the incredibly powerful toxins that define so many of these bacteria.
Yeah, from Bacillus anthracis with its unique protein capsule and that three-part toxin delivery system.
To Clostridium perfringens, just dissolving tissue with its alpha toxin.
And Listeria surviving the cold and invading our cells. It's really a masterclass in microbial adaptation and warfare. The classification scheme, simple as it seems spores, oxygen really does predict a lot about how and where these bugs cause trouble. Spores mean persistence in the environment. Anaerobic means deep tissue infections.
Absolutely. So let's wrap up with a final thought for you, our listeners. Think about those two Clostridium neurotoxins again. Tetanospasmin causing spastic paralysis by blocking the stop signal. Botulinum toxin causing flaccid paralysis by blocking the go signal.
Two toxins from the same genus, achieving completely opposite effects by targeting different sides of the same delicate balance, the balance needed for normal muscle control.
So, considering that knife-edge balance, what does that contrast really tell you about the sheer level of molecular precision required for something as simple as, say, picking up a glass or blinking your eyes, without either constant spasm or complete failure? What stands out to you about that hidden complexity? Something to think about.